Welcome to the Deep Dive. This is where we really dig into complex subjects, look at the research, and try to bring you the core ideas, the real essence of the knowledge. Today, we're exploring something everyone goes through but maybe doesn't fully grasp: how our immune system changes as we age. Picture this, it's probably familiar. Imagine someone, let's say they're 68, reasonably healthy overall. Let's call him Alex. Alex eats pretty well, stays active, has friends. But lately, maybe over the last year, things feel different. They're getting sick more often, colds, flu, whatever it is. It just hangs around longer. And there's this deep fatigue, it wasn't there before. Alex says they just don't bounce back anymore. And well, they're right. It's not just in their head. There are real biological shifts happening inside their cells. So what's actually going on inside Alex's body? What are these complex biological processes that are changing their immune system, making them more vulnerable, slower to recover? Well, the truth is, aging brings these really profound, multi-layered changes to our immunity. We often talk about two key concepts, and they're linked.
There's immunosenescence, that's the sort of gradual decline in immune function. And then there's inflammaging, this chronic low-level inflammation that just kind of smolders away. It's a really complex puzzle. I mean, think about it from the tiny world of your gut microbes all the way down to the stem cells in your bone marrow. A whole range of factors are involved in why an older person's body doesn't quite police itself against infections or diseases or even things like cancer cells as well as it used to. So our mission today in this deep dive is to really unpack the science behind these crucial ideas. We'll start with immunosenescence, how the immune system itself ages. Then we'll tackle inflammaging, that persistent background inflammation. After that, we'll look at the surprising role of the aging microbiome, especially in the gut. And finally, we'll examine something discovered more recently but incredibly important: clonal hematopoiesis of indeterminate potential, or CHIP. We're going to dig into the mechanisms, the whys, and the really big implications of all this, using Alex's experience as our guide, to make it relatable. It's about getting a quick but really thorough understanding, hitting those aha moments where things connect without getting totally overloaded. We're aiming for real depth, but keeping it engaging. Okay, let's dive in. Chapter 1, Immunosenescence, the aging of the immune system.
Yeah, and what's really striking about immunosenescence is that it's not like your immune system just suddenly fails one day. It's much more gradual. It's the slow, complex deterioration. I like the analogy of an aging army. It's not just that you lose soldiers. The whole structure changes. You get fewer new recruits, fresh faces. You have more veterans, sure, but many of them are kind of past their prime, maybe less agile, less adaptable. This fundamental shift in the army's makeup, its overall capability, that's really a key signature of aging.
That's a great analogy.
Yeah.
So what are the biggest changes, the real hallmarks of this aging immune army? Where does it start?
Well, one of the most significant changes, and it starts surprisingly early, is thymic involution, the thymus. It's the small organ behind your breastbone. Its main job, its critical role, is to be the training ground for T cells. T cells are these vital white blood cells. And the thymus educates them, basically teaching them to recognize foreign invaders, viruses, bacteria, without attacking the body's own healthy cells. Self-tolerance, that's key. But here's the kicker. This involution, the shrinking, it starts really early, often in your 20s. By the time someone like Alex reaches 68, their thymus is mostly, fatty tissue. It's largely atrophied. And the impact is huge. It drastically cuts down the production of new T cells, especially what we call naive T cells. These naive cells are the fresh recruits. They haven't seen action yet, haven't encountered a specific germ. They're essential for tackling new infections or responding well to new vaccines. Studies absolutely confirm this, a major drop in primary lymphocyte production in older people. It's due to the thymus shrinking and also changes in the bone marrow where these cells originate. So, in our analogy, the main training academy, the thymus, is slowly winding down production. Fewer adaptable recruits are joining the fight against new threats.
Okay, so if the factory for new T cells is basically shutting down, what does the immune system do? How does it cope with fewer recruits?
It compensates, essentially. Since producing brand new naive T cells and B cells slows right down, the system starts expanding existing clones. These are the immune cells, both T and B types, that have already met specific enemies in the past. They're the veterans. So over decades, you accumulate huge numbers of these memory T cells and memory B cells. They remember past threats, maybe latent viruses like CMV, which we'll talk more about, or common bugs like flu, or even past vaccinations. But the consequence is this big shift in the ratio. Younger people have a good balance of naive and memory cells. older adults, heavily skewed towards memory cells. Someone in the 70s might have blood just dominated by these experienced memory T cells, the veterans, but very few inexperienced naive T cells ready for something new. And that imbalance is a core reason older people have weaker, slower responses to new infections or new vaccines. They struggle to adapt. Plus, even these long-lived memory cells, these veterans, they can get exhausted or dysfunctional over time. They lose their edge, like a soldier whose stamina just isn't what it used to be.
Right, so it's like the immune system is stuck using its old playbook. Good for familiar enemies, but not great for new challenges. Does something similar happen with B cells, the antibody factories? Does their repertoire shrink too?
Yes, it's a very similar story with B lymphocytes, unfortunately. A parallel decline happens there too. Remember, B cells are key for humoral immunity-making antibodies, but with age, B cell production in the bone marrow drops off. This leads to a less diverse pool of B cells overall, and that pool tends to be biased towards remembering infections from earlier in life, so consequently, older folks often show weaker antibody responses when they get a new vaccine or encounter a totally new bug. Their ability to generate fresh, high-quality antibodies is just impaired. And there's another issue. Quality control seems to slip. Aged B cells might actually produce more autoantibodies, the ones that mistakenly attack your own body. At the same time, they produce fewer of the really effective high-affinity antibodies needed to neutralize pathogens properly. It's like immune system wear and tear. So metaphorically, the old playbook is huge, but the ability to write new, effective plays for novel threats is really limited. The memory is vast, but less adaptable.
Okay, so it affects the adaptive specialists, the T and B cells. But what about the first responders, the innate immune system? Does that slow down too?
It absolutely does. The innate system, that rapid, non-specific first line of defense, it also takes a hit with age. Take macrophages, for example. These are crucial cells that engulf pathogens, clean up debris, and present pieces of invaders to T cells to kickstart the adaptive response. Well, with age, macrophages become less efficient at both engulfing things, phagocytosis, and showing those pieces to T cells, antigen presentation. And sometimes they actually lean towards being pro-inflammatory themselves, especially the M1 type found in tissues. They contribute to the problem instead of resolving it.
And other innate cells?
Yeah, like natural killer cells or NK cells. These guys are important for directly killing infected cells or even cancer cells without needing prior instructions. In older people, NK cells often show reduced expression of the receptors they need to recognize and kill targets effectively, so their killing power diminishes. And neutrophils, another key first responder, they get slower at moving to the site of infection that's called chemotaxis, and they're less effective at killing microbes once they get there. So you see, it's across the board. Both the rapid first responders, the innate system, and the targeted special forces, the adaptive system, they all lose some of their edge, their sharpness with age.
This sounds like it connects directly to cellular senescence, right? You mentioned these cells that are alive but dysfunctional. How do they fit into this picture of immunosenescence?
That's a really critical piece of the puzzle. Cellular senescence is fundamental here. It's basically a state where cells permanently stop dividing. They can't replicate anymore, often triggered by things like too many divisions or chronic stimulation from fighting infections over a lifetime or accumulated damage. And what happens is these senescent immune cells, you can identify them like T cells that lose a marker called CD28 and gain one called CD57. They really accumulate in older adults. And like you said, they're alive but dysfunctional. They don't respond to new threats. They functionally inert when something new comes along. But crucially, they're not quiet bystanders. They actively pump out a whole cocktail of pro-inflammatory molecules, cytokines, chemokines, enzymes that break down tissue. This is called the senescence-associated secretory phenotype, or SASP, and this links directly to our next topic, inflammaging. Because these senescent immune cells, through their SASP, are constantly adding fuel to that chronic, low-grade inflammation we see in aging. They're like disgruntled retirees who won't stop shouting inflammatory messages.
Okay, let's just pause and unpack that. We've seen the immune system aging out in multiple ways. Fewer new T cells, less diverse B cells, slower innate responses, and now these senescent cells actively causing trouble. So for Alex, our 68-year-old who doesn't bounce back and gets sick often, what's the bottom line impact?
Well, the crucial thing to grasp is that this isn't just a passive fading away. It's an active remodeling process. And it leaves the whole immune system less flexible, often stuck reacting to old threats and just fundamentally less prepared for new ones. For Alex, it means their system is slower off the mark when they encounter, say, a new flu strain or a bacterial infection they haven't seen before. Their functional immune reserve is just lower. They can still fight things off, definitely, But the point at which their system gets overwhelmed comes much sooner than it did when they were younger.
So that explains the frequent illnesses, the lingering symptoms.
Exactly. Higher susceptibility to infections, they get sick more often, and it's often more severe, lasts longer. Weaker vaccine responses, too. That standard flu shot might not give them the same protection it gives a 30-year-old. That's why we have those high-dose or adjuvanted vaccines for older adults. And another critical point, often missed, is cancer surveillance. A less effective immune system is less good at spotting and eliminating rogue cancer cells early on. So cancer rates are higher in older people, partly due to this immunosenescence. So it's not just decline, it's this active detrimental shift. And this whole picture of a remodeled, less flexible, somewhat exhausted immune system leads us directly to the next critical piece, this chronic inflammatory state that often comes with aging. We call it inflammaging.
Right. It makes perfect sense. It's not just weakness. It's a kind of counterproductive overreaction, too. Which brings us squarely to Chapter 2. Inflammaging the chronic inflammatory state of aging.
Exactly. Inflammaging. It's this really pervasive, low-grade chronic inflammation. And it's there even when there's no obvious infection. It's like a low fire constantly smoldering under the surface. Clinically, we see it as slightly but persistently elevated levels of certain pro-inflammatory markers in the blood. Things like IL-6, interleukin-6, IL-1 beta, TNF-alpha, tumor necrosis factor alpha, and also C-reactive protein, CRP. Even slightly high levels of these indicate this constant low-level immune activation, and the significance is huge. We now know this chronic inflammation is a major risk factor for a whole host of age-related diseases. It's implicated in arthritis, atherosclerosis, the hardening of arteries leading to heart attacks and strokes, also type 2 diabetes, Alzheimer's, various cancers, and just general frailty. Inflammaging is really a foundational problem in aging.
So it's not just a side effect of getting older, it's actively driving these diseases. What's actually causing this chronic fire, this pile up of inflammatory noise, as you put it?
It's definitely multifactorial. There's no single cause. First, we need to expand on the accumulation of senescent cells we mentioned. It's not just immune cells. Senescent cells build up throughout the body's tissues in fat, artery walls, the liver, skin, everywhere. And remember, they're not just sitting there. They have that senescence-associated secretory phenotype, SASP. They're constantly spewing out that inflammatory cocktail cytokines like IL-6, IL-1 beta, chemokines, tissue-damaging enzymes. As these cells pile up with age, their SASP creates this local inflammatory environment, which then spills over and contributes to systemic inflammation body-wide. Think of it like a growing number of damaged, retired cells that just won't shut up, constantly yelling inflammatory signals and polluting the whole system.
Okay, so these senescent cells are actively fanning the flames. What else is adding fuel to this fire? You mentioned the body's own garbage.
Yes, absolutely. Another key driver is the buildup of self-debris, or cellular garbage. Over decades, our normal metabolic processes and cell turnover generate a lot of waste products. Things like oxidized fats, misfolded proteins that clump together, bits of dead cells, DNA fragments floating around. In youth, our body's cleanup systems, like autophagy, the cell's own recycling system, are pretty efficient at clearing this stuff out. But with age, these clearance mechanisms become less effective, so the self-debris hangs around longer. And this debris acts like danger signals. We call them DAMPs, danger-associated molecular patterns. They constantly provoke the immune system, keeping it low-key activated. Think of cholesterol crystals in artery plaques or protein aggregates in the brain in Alzheimer's. These things continuously irritate immune cells. When the body's cleanup crew slows down, the lingering trash keeps the immune system slightly on edge, maintaining that low-level alert. That's a really clear image, the cleanup crew falling behind. Right.
What about the impact of fighting infections throughout life? Does that cumulative burden contribute?
Oh, absolutely. It's a very significant factor. Chronic infections and the persistent antigenic load from a lifetime of exposures play a huge role. And one of the main culprits suspected of driving both immunosenescence and inflammaging is cytomegalovirus, or CMV. It's an incredibly common herpes virus. Most adults carry it latently. It doesn't usually cause problems, but it periodically reactivates at a low level, subtly stimulating the immune system. The issue is a huge chunk of an older person's T-cell army might be dedicated just to keeping this one virus in check. And ironically, many of those CMV-specific T-cells become senescent themselves. Studies consistently link being CMV positive to having higher inflammatory markers, especially IL-6, and even higher mortality risk in the elderly. But it's not just CMV. Repeated acute infections over a lifetime or chronic low-grade infections like persistent gum disease, periodontitis, or reactivations of other latent viruses, they all provide this constant antigenic burden. It keeps the immune system simmering, sustaining this smoldering inflammation. It's like a low-intensity battle that never fully ends, just wears the system down.
And what about fat tissue? We usually think of it as just storage, but you mentioned it's active. How does fat contribute to inflammation as we age?
That's a really important and maybe surprising point. Adipose tissue, or body fat, is far from passive. It's actually a very active endocrine organ. It secretes lots of hormones and inflammatory mediators, things like IL-6, TNF-alpha, leptin. And as people age, they often tend to accumulate more visceral fat, the fat around the internal organs, even if their overall weight doesn't change much. This aged fat tissue itself becomes more inflammatory, it accumulates more senescent cells, gets infiltrated by pro-inflammatory macrophages. In fact, obesity is known to significantly accelerate inflammaging. Sometimes it's called metaflammation because of the metabolic links. Conversely, look at centenarians, people living to 100 or more. They often have remarkably low levels of inflammation despite their age. It highlights the connection. This fat-driven inflammation contributes to insulin resistance, type 2 diabetes risk, and also vascular inflammation, feeding directly into atherosclerosis.
Wow, even fat cells are in on the act. And the gut microbiome, it seems connected to everything. How does it play into inflammaging?
Indeed, the gut microbiome is another absolutely critical piece of the inflammaging puzzle, and one that's gaining enormous attention. This vast ecosystem of trillions of microbes in our intestines plays a massive role in tuning our immune system and controlling inflammation body-wide. And just like everything else, the microbiome changes significantly with age, usually for the worse. In young, healthy people, you have a diverse, balanced microbiome. It helps train the immune system properly and maintains the integrity of the gut lining, that crucial barrier. But as we get older, microbial diversity often plummets. The balance shifts. You tend to lose beneficial species and gain more pro-inflammatory ones. This imbalance is called dysbiosis.
And this leads to leaky gut.
Exactly. Older individuals often experience increased intestinal permeability or leaky gut. This allows bacterial components, things like LPS or endotoxin, bits of bacterial cell walls, to leak out of the gut and into the bloodstream. This constant low-level trickle of microbial stuff into the circulation acts as a potent trigger for chronic immune activation throughout the body. It directly fuels inflammaging, so an aging, out-of-balance gut microbiome combined with the leaky gut barrier becomes this major persistent source of inflammation, driving the problem from within. We'll get into some striking experiments on this soon.
Okay, here's where it gets really interesting. So it's not just that the immune system weakens, but it's also constantly on high alert, causing damage. It's like the body's fighting a perpetual low-level war from within. So what else is contributing to this inflammatory overdrive?
Indeed. If we connect this to the bigger picture, inflammaging isn't a single cause, but a complex tapestry woven from senescent cells, lingering cellular debris, chronic infections like CMV, inflammatory fat, and that shifting gut microbiome. All these factors interact, often in vicious cycles. Senescent cells and fat make IL-6, that raises systemic IL-6, the liver makes more CRP, and high CRP predicts frailty and disease. It all feeds back. The result is that older people tend to have this higher baseline inflammatory tone compared to when they were young, even if they feel perfectly healthy. And the consequences are real. This chronic inflammation damages tissues over time, artery walls, brain cells, and paradoxically, it can mess up the acute immune response when you actually need it, causing feedback inhibition or exhausting immune cells. For Alex, our patient, this might show up as high hsCRP or IL-6 on blood tests. Large studies show these markers are powerful predictors of disability and mortality risk in older adults. Low levels correlate with successful aging. So the implication is huge. Tamping down inflammaging could significantly improve health span. Claudio Franceschi, who coined the term, described it brilliantly. The immune alarm never fully turns off in old age. It's constantly on, wearing things down. That's why fighting inflammaging through drugs, diet, maybe senolytics is such a hot area of research right now. And building on this idea of internal drivers, we now understand a hidden phenomena in the bone marrow that acts as a significant accelerant, clonal hematopoiesis.
That's incredible. So the very cells that are supposed to protect us can, with age and certain mutations, become agents of inflammation. This raises an important question. What other seemingly distant parts of our body might be contributing to this constant alarm state?
Indeed. If we connect this to the bigger picture, CHIP is a profound insight into how aging at the stem cell level can trigger systemic inflammation. But the body is an incredibly interconnected system, and one of the most surprising and impactful drivers of both immunity and inflammation, especially in aging, actually resides in our gut, the microbiome.
Right. CHIP. Clonal hematopoiesis. It sounds technical, but you're saying it's a major piece linking aging, bone marrow, and inflammation. Let's dive into Chapter 3. Clonal hematopoiesis, CHIP, and inflammation.
It is technical, yeah, but the implications are really profound. CHIP stands for clonal hematopoiesis of indeterminate potential. Essentially, it refers to acquiring specific DNA mutations as we age, but specifically within our hematopoietic stem cells, HSCs. Remember, HSCs are the master stem cells in the bone marrow. They give rise to all our blood cells, red cells, white cells, platelets. Everything starts there. As we get older, these HSCs can pick up spontaneous mutations, often in specific genes like TEA2, DNMT3A, ASXL1, or JAK2. Now, if one of these mutations gives that particular stem cell a growth advantage, it starts dividing more than its neighbors. Over time, the descendants of that single mutated stem cell can expand and take over a significant fraction of blood production. So you end up with a large population of blood cells all carrying that same mutation, all from one clone. It's kind of like a lottery in the bone marrow. A few stem cells get a lucky mutation that lets them outcompete the others, and their offspring eventually dominate the blood pool.
So, a mutation in a master stem cell leads to a dominant clone of blood cells. It sounds like a step towards cancer, doesn't it? Or is that the initial thought?
That's exactly how it was initially understood, yes. CHIP was first recognized mainly as a risk factor for blood cancers, especially myeloid leukemias like AML. That's why it's called indeterminate potential. It's not cancer itself, but it's a potential precursor state. Though the actual risk of progressing to leukemia is pretty low for most people, maybe 0.5 to 1% per year. But then came the really big surprise, the aha moment from later research. Scientists found CHIP has a much broader and arguably more significant impact. CHIP dramatically increases risk for cardiovascular disease and overall mortality, primarily driven by heart problems, not just that small cancer risk. The numbers are quite staggering. Having a CHIP mutation is associated with roughly a doubling of risk for coronary artery disease, things like heart attacks. It's also linked to stroke and heart failure. One major study found CHIP carriers had almost twice the risk of heart disease and an astonishing 4-fold higher risk of having an early heart attack compared to people their age without CHIP. Just think about that effect size. It's huge. It's on par with, or even greater than, many classic heart risk factors like high cholesterol or smoking.
Wow. That is a massive, unexpected finding. CHIP isn't just about a small blood cancer risk, it's a major hidden driver of heart disease. But how? How does a mutation way down in a bone marrow stem cell cause problems in arteries? What's the inflammation connection?
That's the absolute key link, and it ties right back into inflammaging. It turns out the mutated blood cells produced by these CHIP clones, especially certain mutations, are inherently more inflammatory. They secrete more inflammatory signals, and that's what accelerates atherosclerosis, the plaque buildup in arteries. Let's look at specifics. Take TT2 mutations. TT2 is a gene involved in regulating DNA methylation, which controls how genes are expressed, especially in immune cells like macrophages. Experiments clearly show that macrophages carrying TET2 mutations are hyperinflammatory. They pump out way more IL-1 beta, IL-6, and other inflammatory cytokines. And these cytokines directly fuel plaque formation and make plaques less stable, more likely to rupture and cause a heart attack. Mouse models where TT2 is knocked out in immune cells develop much worse atherosclerosis purely because their white blood cells were so inflammatory.
So the mutation makes the immune cells themselves more aggressive.
Exactly. And it's similar for other CHIP mutations. The JAK2 V617F mutation is another common one. It also ramps up inflammatory cytokine production, and it can make blood clot more easily, adding to the risk. It might carry the highest heart risk of all CHIP mutations. DNMT3A mutations also create a pro-inflammatory state in monocytes and macrophages, the cells that crawl into artery walls and drive plaque growth. So essentially, CHIP clones effectively turn immune cells into inflammation amplifiers. The leading idea from researchers like Jaiswal and Libby is that CHIP might be a major reason why simply getting older is such a strong risk factor for heart disease. Older people are much more likely to have CHIP, maybe 10-20% over age 70. So these stealth mutations accumulating over time could literally make your immune cells hot-headed, constantly promoting inflammation that damages your arteries.
Hot-headed immune cells. That's a striking image. So these rogue clones are adding fuel to the inflammatory fire from within. What does this mean clinically? If someone is found to have CHIP, what do we do? Should everyone get tested?
That's the big clinical question right now, and it's still evolving. Currently, there's no standard recommendation for routine CHIP screening in the general older population. The testing requires specialized DNA sequencing of blood, and crucially, we don't have a direct therapy to just eliminate the CHIP clone itself. However, experts are starting to suggest testing in certain high-risk situations, like maybe an older person who has severe heart disease unusually early without the typical risk factors, or someone with unexplained persistent abnormalities in their blood counts, like mild anemia.
Okay, so selective testing for now. But if CHIP is found, how does it change things?
It definitely changes the risk assessment and management strategy. If CHIP is found, clinicians should absolutely manage cardiovascular risk factors much more aggressively. Knowing about CHIP provides strong justification for that, given the doubled heart disease risk and higher overall mortality. So that means really tight LDL cholesterol control, doubling down on anti-inflammatory lifestyle advice, diet, exercise, and potentially considering drugs like low-dose aspirin or statins more readily. Statins, besides lowering cholesterol, also have anti-inflammatory effects on plaques, which is highly relevant here.
And could knowing the specific mutation guide treatment even more?
Potentially, yes. That's where future therapies might come in. Knowing someone has a TE2 or IL-6-related mutation could eventually guide targeted anti-inflammatory treatment. Remember the CANTOS trial? It tested an IL-1 beta blocker, canakinumab, after heart attacks. A later analysis showed that patients in that trial who also had TP, especially TT2 mutations, got a much bigger benefit from blocking IL-1 beta. That was huge. It validated the idea that inflammation is the key link for Phi theta's cardiovascular effects. It strongly suggests that anti-cytokine drugs targeting IL-1 or IL-6 might be particularly effective for mitigating risk in CTP carriers. Clinical trials are actually underway now to test this directly.
Okay, so for now, finding THAP is basically a red flag.
Exactly. It's a significant red flag. If it's found, that person needs close monitoring for both heart disease progression and any signs of blood cancer development, though that's less common. And their risk factors need intensive management. Referral to clinical trials or a hematology consult might be considered too. But remember, CHP itself usually has no symptoms. It's often found by chance. And most people with CHP won't get leukemia. The bigger issue for most is the cardiovascular inflammation. So for Alex, our 68-year-old, maybe they have CHC, maybe not. But if they did say a TET2 mutation found, incidentally, it could partly explain their fatigue. Inflammation causes fatigue. And would absolutely mean we need to be hypervigilant about preventing heart disease and stroke.
So the very cells that are supposed to protect us can, with age and certain mutations, become agents of inflammation. This raises an important question. What other seemingly distant parts of our body might be contributing to this constant alarm state?
Indeed. If we connect this to the bigger picture, CHIP is a profound insight into how aging at the stem cell level can trigger systemic inflammation. But the body is an incredibly interconnected system, and one of the most surprising and impactful drivers of both immunity and inflammation, especially in aging, actually resides in our gut. The microbiome.
Right, the microbiome seems to be everywhere in health discussions now.
Yeah.
So let's move to Chapter 4, the aging microbiome and its influence. How does our gut connect to Alex's fatigue and immune issues?
Yeah, understanding Alex's situation, or aging immunity in general, really requires a look at the gut microbiome. It's this incredibly complex community of trillions of microbes, bacteria, fungi, viruses living in our intestines. And it plays an absolutely crucial role in shaping our immune system and controlling our overall inflammatory state. Like other body systems, the microbiome changes significantly as we age, and usually these changes aren't for the better. In youth, a diverse, balanced microbiome helps train the immune system, promotes tolerance, and crucially maintains the integrity of the gut, lining the barrier between the gut contents and the rest of the body. But in older age, microbial diversity tends to drop. The proportions of different bacterial groups shift, often leading to more pro-inflammatory species taking over and fewer beneficial ones. This imbalance is called dysbiosis.
And that leads to the leaky gut phenomenon we hear about.
Exactly. Older individuals often experience increased intestinal permeability or leaky gut. The gut barrier becomes less effective. This allows bacterial products, things like LPS, also called endotoxin, or other microbial fragments, to leak from the gut into the bloodstream. This constant low-level leakage, this translocation, triggers chronic immune activation systemically. It's a major source fueling inflammaging. So an aging dysbiotic gut, combined with a leaky barrier, is a significant driver of that chronic smoldering inflammation we've been talking about. It fuels it from the inside out.
That's incredible, the idea that what's living inside our gut could literally be making us older or younger.
Yeah.
What's the most striking evidence for this gut-aging connection?
Some of the most compelling, really eye-opening evidence comes from fecal microbiota transplant, or FMT experiments, mostly in animal models so far. FMT is basically transferring gut microbes from one animal to another, and the results in aging studies have been pretty dramatic. Take the young to old FMT studies. When researchers transfer gut microbes from young, healthy mice into old mice, the old mice often show remarkable health improvements, suggesting systemic rejuvenation. For instance, a 2022 study showed that giving aged mice microbes from young mice actually reversed certain hallmarks of aging, not just in the gut, but also in the brain and even the eyes. The aged mice showed less brain inflammation, their gut barrier function improved, and their retinal health got better. It was quite widespread. Another study found old mice given young microbiota actually lived longer and had better immune function than control old mice. So, quite literally, young poop made old mice younger in several measurable ways. It strongly suggests these microbes have powerful systemic effects.
That is truly astounding. So does it work the other way too? Can transplanting an old microbiome make a young animal age faster?
Excellent question. And yes, the inverse experiment is perhaps even more striking as proof of concept. Transferring an aged microbiome into a young host can actually cause premature aging-like effects. Young mice that received gut contents from old mice quickly developed signs of leaky gut, systemic inflammation, and even cognitive decline compared to young mice getting microbes from other young mice. So essentially, the old gut flora made the young mice act older. It's a powerful demonstration that the microbiome isn't just a bystander. It can directly drive aging phenotypes. Similar findings have been seen in other models too, like short-lived killifish, where young microbiota expanded lifespan, and even fruit flies. It seems to be a conserved biological principle.
So how exactly do these gut microbes exert such a powerful influence on aging and immunity? What are the mechanisms?
Well, the main idea is that an unhealthy, aging-associated dysbiotic microbiome actively promotes age-related disease, while a youthful, diverse one might be protective or even rejuvenating. The how is multifaceted. Firstly, gut bacteria are metabolic powerhouses. They produce countless metabolites from digesting our food, especially fiber. Things like short-chain fatty acids, butyrate, propionate, acetate are incredibly important. They directly influence immune cell function and inflammation, both locally in the gut and systemically. Butyrate, for example, is generally anti-inflammatory. Secondly, there's the gut-brain axis. Gut microbes communicate with the brain, influencing neurological health, neuroinflammation, and potentially cognitive function in aging. And third, as we discussed, they critically influence gut barrier integrity. A healthy microbiome helps keep the barrier strong, preventing that inflammatory leakage. An unhealthy one contributes to leakiness, so they control that constant trickle of inflammatory signals from the gut.
Okay, this is really fundamental. So for Alex, our 68-year-old, how might their specific microbiome be contributing to their fatigue and immune issues? And what about potential therapies, even if experimental?
It's very plausible that Alex's microbiome is playing a role. Maybe years of a standard Western diet, maybe some antibiotic courses. These things can shift the microbiome towards a less healthy state. This could directly contribute to their higher baseline inflammation and maybe lower their resistance to infections. Perhaps they have fewer beneficial lactobacilli or bifidobacteria, for example. We're seeing more evidence linking specific microbial patterns in the elderly to frailty or higher inflammation levels. And this, of course, points to the really tantalizing potential of microbiome modification as a therapy. Imagine being able to restore a youthful microbiome in an older person, maybe through FMT from a healthy young donor. The animal studies suggest it could have significant benefits. And this is but what about humans? This is where we need extreme caution. Right now, there's no approved medical use of FMT for general anti-aging in humans. It's highly experimental for that purpose. While the animal data is exciting, we're still waiting for solid human clinical trial data showing it's both safe and effective for improving health or slowing aging markers in people. It carries real risks if not done properly. So while researchers are definitely pursuing this, maybe someday we'll have proven microbiome rejuvenation therapies, but we're not there yet for routine use.
And that brings up a really critical point about safety. With such exciting potential shown in animals, people might be tempted to try things themselves, right?
Absolutely. And this is an extremely important warning we have to give. While the science is fascinating, DIY fecal transplants are incredibly dangerous and strongly discouraged. There have been documented cases of serious, life-threatening infections and even deaths from FMT using improperly screened donor stool or done outside regulated medical settings. It's not a simple procedure. The FDA, for instance, only approves FMT for recurrent C. diff infections, and explicitly warns that unsupervised FMT getting stool from unscreened donors using online treatments can cause potentially fatal infections. So to anyone listening who might think about trying this at home, please do not do it. It needs rigorous study, controlled conditions, and medical oversight before it could ever be considered safe or effective for anti-aging.
Okay, very clear warning. So what can people do safely today to support a healthy microbiome for better immune resilience?
The practical steps really come down to diet and lifestyle. Eating a diet consistently high in diverse fiber from a wide variety of plant foods is key. Fiber feeds beneficial bacteria and encourages diversity, leading to healthy metabolites like butyrate. Some studies suggest specific probiotics or prebiotics might help reduce inflammation or improve gut health in older adults, but the evidence is still evolving for specific strains. And basic things. Avoiding unnecessary antibiotics, which can really damage the microbiome, and maintaining good oral hygiene, as oral bacteria can affect the gut, are always good advice. In essence, your gut microbiome can either fan the flames of inflammaging or help put them out. Nurturing a healthy gut environment is a cornerstone of immune resilience as we age.
That's incredible, the idea that what's living inside our gut could literally be making us older or younger. It really highlights how interconnected everything is. But beyond the gut, what about the very foundational cells of our body, like stem cells and the environment they live in?
Exactly. If we connect this to the bigger picture, the microbiome's influence is a powerful example of how systemic factors, even beyond our direct genetic code, drive aging. This concept of systemic influence, of circulating factors affecting our entire body, was profoundly revealed through some classic experiments looking at stem cells and their niche.
Right, then niche. So this takes us to Chapter 5, Stem Cells, Niche, and Rejuvenation Parabiosis Lessons. We've talked about hematopoietic stem cells, HSCs, aging and mutating, but what about their home, their environment?
Precisely. We need to consider not just the HSCs themselves, but their niche, the specialized microenvironment within the bone marrow that supports and regulates them. And just like the stem cells, this niche also undergoes significant changes with age.
How does the niche change?
Yeah.
And how does that affect the stem cells and our immunity?
One really prominent change is marrow adiposity. You know the saying, our marrow runs to fat as we age. It's quite literal. Older bone marrow accumulates more fat cells, adipocytes. It shifts from red marrow, which is active in blood production, towards yellow marrow. And these fat cells aren't just passive filler, they actively change the local environment. They secrete fewer supportive factors that HSCs need and more pro-inflammatory signals like certain cytokines. This altered environment tends to skew blood production away from lymphocytes, T and B cells, and towards myeloid cells like monocytes, neutrophils, which can contribute to inflammation. The niche becomes less supportive for adaptive immunity and more pro-inflammatory. We find higher levels of IL-6 and other inflammatory cytokines in old marrow. Another key change happening not just in marrow but throughout the body is fibrosis and increased stiffness. Aging tissues, including the bone marrow sometimes, can develop fibrosis and excess buildup of extracellular matrix proteins like collagen. This physically changes the tissue. More generally, the extracellular matrix, ECM, the scaffold around our cells, gets stiffer with age everywhere. This happens due to things like cross-linking of collagen fibers. And why does stiffness matter? Because the mechanical properties of tissues profoundly influence cell behavior. A stiff environment can actually make cells more inflammatory and pro-fibrotic. It creates a vicious cycle. Stiffness leads to cell activation, which causes inflammation and more fibrosis, which makes the tissue even stiffer. We see this in aging skin, lungs, blood vessels. This stiffness itself can trigger inflammatory signaling pathways inside cells, things like APTZ or TGF-beta. So the very physical mechanics of older tissues can help sustain chronic inflammation and hinder regeneration. It's a fundamental physical change driving age-related decline.
Wow, the idea that tissue stiffness itself fuels inflammation is mind-bending. But then there are those famous parabiosis studies suggesting systemic factors in blood can actually reverse some aging effects. Can you walk us through those?
Yes, the parabiosis experiments. They're really a cornerstone of modern aging research, providing dramatic proof of concept for systemic factors. Parabiosis is this experimental setup where two animals, usually mice, are surgically joined so they share a blood circulation. In the classic studies, they joined a young mouse and an old mouse. And the results were stunning. The old mouse showed clear signs of rejuvenation in multiple tissues, better muscle repair, improved heart function, even better cognitive function. Conversely, the young mouse often showed signs of accelerated aging. This was powerful evidence that factors in young blood can rejuvenate old tissues, and/or factors in old blood can actively harm young ones. It really kicked off the hunt for these circulating elixirs of youth, or factors of aging.
So what factors were identified? Were they successful in finding the elixir?
Well, researchers have identified several candidate factors. GDF11 was a big one for a while, thought to rejuvenate muscle and heart, though there's been some debate and complexity around it since. Other potential youth factors like oxytocin for muscle regeneration were found. On the flip side, things like RANTES, CCL11, were identified as potential pro-aging factors in old blood linked to cognitive decline. More generally, it's clear that old blood has higher levels of pro-inflammatory cytokines and factors that inhibit cell growth, while young blood has more supportive factors. But this led to a lot of, hype about young blood. Some clinics even started offering young plasma infusions, which raised huge ethical concerns and lacked scientific backing. It's really crucial to state. There is currently no proven anti-aging benefit from young blood transfusions in humans. Small trials, like giving young plasma for Alzheimer's, haven't shown convincing results. Safety and effectiveness in humans are far from established.
Okay, so no vampire therapy just yet. But what's the big takeaway lesson from parabiosis then?
The truly profound lesson is that aging is malleable, and systemic factors matter immensely. It's not just about our genes ticking away inevitably. The fact that old tissues could be rejuvenated when exposed to a young systemic environment was revolutionary. It showed aging isn't entirely fixed. This gives real hope that by modulating the blood environment, maybe removing harmful factors or adding beneficial ones, we might genuinely improve health and function in older age. It shifts aging from being seen as inevitable decline to a potentially modifiable biological process.
So if we can't connect Alex to a young person, what are the practical strategies being explored now to mimic those parabiosis effects or fix the aging niche?
Right, that's the billion-dollar question. How do we translate this? We can't do parabiosis in people. But several alternative strategies are under active investigation. One very exciting area is senolytics. These are drugs designed to selectively kill off those senescent cells we talked about. By clearing out this inflammatory deadwood, the hope is to reduce the SASP, reduce fibrosis, and generally improve the tissue environment. Early human trials, like using dasatinib plus quercetin for lung fibrosis, have shown some encouraging hints. It's plausible senolytics could make old niches, like the bone marrow, healthier. Another approach is targeting fibrosis and stiffness directly with antifibrotic drugs or ECM modifiers, trying to soften tissues by blocking pathways like TGF-beta or breaking down crosslinks. Still mostly experimental, but a key area of anti-aging research. Then there's the idea of modulating the niche with specific growth factors, delivering beneficial factors, maybe like GDFs or others, to try and replicate the positive effects seen in parabiosis without needing the young blood itself. A more targeted molecular approach. And interestingly, there's research into therapeutic plasma exchange. The TRIM plasma trial is testing whether simply removing some of an older person's plasma and replacing it with selenialbumin, essentially diluting out the bad stuff, can improve health markers. The idea there is that removing pro-aging factors might be more effective than adding youth factors. It's a very active area, moving from animals to humans.
So lots of possibilities on the horizon, but clearly still early days.
Very early days for most of these in terms of proven human benefit. We absolutely need to avoid the hype. The promise is that aging is modifiable, but people should definitely steer clear of unproven young blood treatments or supplements sold outside rigorous trials. They lack evidence and could be harmful. For Alex, while direct rejuvenation is sci-fi for now, this research suggests future therapies might target these systemic factors.
Wow, we've covered so much ground already. From the shrinking thymus to mutated stem cells and even the gut. It's clear that aging impacts immunity on multiple interconnected levels, often leading to this chronic inflammatory state. Let's take a moment to recap the core insights we've uncovered so far.
Absolutely. If we connect this to the bigger picture, what we're seeing is a complex interplay that fundamentally alters the body's ability to police itself. Here are the key points to carry forward, almost like a mental flashcard set. First, immunosenescence. The immune system ages. Fewer new cells due to thymic involution B-cell decline, more veteran memory cells. Result: less adaptable to new threats. Second, inflammaging, that persistent low-grade inflammatory state, driven by senescent cells, SASP, cellular debris, chronic infections like CMV, inflammatory fat, and a shifting gut microbiome. This constant alarm fuels many age-related diseases. Third, CHIP, clonal hematopoiesis, age-related mutations in blood stem cells like TT2, DNMT3A, JAK2, creates rogue inflammatory blood cell clones, significantly boosts cardiovascular and mortality risk, a hidden accelerant. Fourth, the aging microbiome. Gut microbial diversity drops with age, often becoming more pro-inflammatory. A leaky gut allows bacterial products to fuel systemic inflammation. Animal FMT studies showed dramatic rejuvenation or aging effects. And fifth, systemic rejuvenation, parabiosis lessens. Experiments proved systemic factors in blood influence aging. Young blood can potentially rejuvenate old tissues and vice versa. Shows aging is modifiable. Interventions like senolytics or plasma exchange hold promise but need rigorous study.
That's a fantastic comprehensive summary. It really nails the complexity and interconnectedness. Okay, now that we understand how the immune system ages and gets inflamed, let's shift to protection. Chapter 6, Vaccination and Immune Interventions in Older Adults. Given this aging immune army, how do we best defend older adults like Alex?
Yeah, this is a huge practical challenge. We know standard vaccines often don't work as well in older adults because of immunosenescence and inflammaging. Take the regular flu shot. It gives decent protection in younger folks, but its effectiveness is definitely lower in seniors. Their immune systems just don't mount as strong a response. So, to counter this, scientists have developed specific enhanced vaccine strategies designed for older populations.
So it's not just getting any vaccine, but the right formulation for their age. What are some of these enhanced strategies?
One straightforward but effective approach is using higher antigen doses. Basically, put more of the active ingredient, the antigen, into the shot. The idea is to give the sluggish older immune system a stronger signal to respond to. The classic example is the high-dose influenza vaccine, like Fluzone high dose. It has 4 times the antigen of the standard shot, and studies consistently show it leads to higher antibody levels and better protection against flu illness in people 65 and older. The new RSV vaccines for older adults also use strong doses for the same reason, to overcome that baseline immunosenescence. Another really important strategy is using adjuvants. These are substances added to vaccines specifically to boost the immune response. They act like an amplifier, sending a danger signal to wake up the immune system and make it react more strongly to the vaccine antigen. The Shingrix shingles vaccine is a great example. It uses a potent adjuvant called S1E09. This provokes a much stronger and more durable T-cell and antibody response than the older shingles vaccine ever did. That's why it's so much more effective. Similarly, the adjuvanted flu vaccine, Fluad, uses an adjuvant, MF59, to help stimulate a better reaction in seniors. Now, the trade-off can sometimes be more local side effects, a sore arm, maybe some redness, but the benefit in terms of improved protection against serious disease is significant.
Makes sense. Give the system a stronger wake-up call. What about the timing? Can changing the number of doses or the schedule help?
Yes, absolutely. Optimized vaccination schedules are another tool in the toolbox. Sometimes giving more doses or spacing them differently works better for older immune systems. For example, older adults often need a 2-dose initial series for some pneumococcal vaccines to get good coverage. Studies on things like hepatitis B vaccines in older or immune-compromised people show better results with double doses or extra shots. And we saw it with COVID-19 too. Boosters were recommended more frequently for older individuals, acknowledging that their immunity might wane faster.
And beyond these current strategies, what's on the research horizon for vaccines specifically for aging?
There's a lot of exciting work. Novel vaccine concepts are being explored. For instance, research into a CMV vaccine. If we could prevent CMV infection, maybe we could lessen some of that CMV-driven immunosenescence and inflammaging burden. That's a big goal. Also, therapeutic vaccines, like cancer vaccines, are being designed specifically with the older immune system in mind. Sometimes they incorporate things like checkpoint inhibitors or cytokines to try and overcome T-cell exhaustion. Still early days for many of these, but promising directions.
Okay. Vaccinations are key, but what about other things? Are there medications or therapies that might actually rejuvenate or boost immune function in older adults?
This is where we get into immune rejuvenation therapies or geroprotective drugs. It's a really hot area. One of the most studied approaches involves low-dose MTOR inhibition, using drugs like rapamycin or its cousins, rapalogs. MTOR is the central pathway controlling cell growth, metabolism, and aging. Now, paradoxically, while high doses suppress immunity, used in transplants, carefully calibrated low doses seem to enhance certain immune functions. A key trial by Manek and colleagues gave healthy older adults a low-dose MTOR inhibitor. Their response to the flu vaccine improved significantly, about 20% better than placebo. They also saw a modest reduction in senescent T cells. Another trial with a similar drug, RTB101, showed fewer respiratory infections in elderly patients taking it. So it seems like gently tuning down MTOR signaling can rejuvenate parts of the immune system, maybe by boosting autophagy, cellular cleanup, and improving T cell function. It's the delicate balance, the Goldilocks dose. Too much suppression is bad, but just the right amount seems beneficial for immune surveillance and vaccine response. This research is really driving biotech efforts to create immune rejuvenating pills. Maybe something older people could take before flu season to boost vaccine effectiveness.
Interesting. What about other drugs like metformin?
Yeah, metformin, the common diabetes drug, is also getting a lot of attention for potential anti-aging and immune effects. Some observational studies hinted at better COVID outcomes in metformin users, but we need more definitive trials. It can reduce inflammation and affect innate immune cells. The big TAME trial targeting aging with metformin should give us clearer answers soon. But honestly, we shouldn't overlook the basics. Vitamin and nutrient supplementation, especially to correct deficiencies. Vitamin D deficiency is incredibly common in older adults and linked to infection risk. Fixing that is real low-hanging fruit. Zinc is crucial for T and B cells. Even mild deficiency can impair immunity. Supplementation can help if levels are low. And critically, protein. Low protein intake worsens muscle loss, sarcopenia, and directly impairs immunity, antibody production, wound healing. Older adults often need more protein, maybe 1 gram per kilogram of body weight per day, or aiming for 25-30 grams per meal to maintain muscle and immune function, plus adequate calories and micronutrients from fruits and veggies. There's research on other things like JAK inhibitors or IL-7 therapy, but they're mostly experimental with significant side effects, not ready for general use in healthy aging. Checkpoint inhibitors used in cancer are too risky due to autoimmunity.
So really, for Alex today, the main actionable thing is vaccines.
Absolutely. The most impactful thing today is for older adults to stay absolutely up to date on recommended vaccines using those age-appropriate formulations. So for Alex, annual flu shot, high dose or Juvenibd, the Shingrix series, pneumococcal shots, COVID boosters, and the new RSV vaccine if eligible. These vaccines dramatically cut the risk of serious infections that their aging immune system would struggle with. And clinically, doctors are often quicker to treat infections aggressively in older patients, maybe starting Tamiflu earlier for flu, or using antivirals monoclonals for COVID more readily than in a younger person.
That makes sense. Proactive protection and quicker treatment. But how do we actually measure someone's individual immune age? Are there tests for that? Let's talk about Chapter 7, biomarkers of immune aging.
Yeah, that's a key question in the field. How do we quantify this? How old is someone's immune system, really? It can vary a lot from their chronological age. Researchers and increasingly clinicians are using a panel of biomarkers to get a handle on immunosenescence and inflammaging. One classic marker is cytomegalovirus, CMV serostatus. Whether you're CMV positive or negative, being positive is often seen as adding to your immune age burden because CMV drives that expansion of late-stage, often senescent T cells we talked about. A CMV positive older adult usually has more of those terminally differentiated CD8 plus T cells, the CD28, CD57 plus ones, and fewer naive T cells than someone CMV negative. Some researchers even defined an immune risk profile in the very old things like being CMV positive plus having an inverted CD4.CD8 ratio, which strongly predicted mortality in studies like the Swedish Octanona ones. Clinically, we don't treat asymptomatic CMV, but knowing the status helps interpret the overall immune picture.
What about measuring the diversity of immune cells directly?
Yes, that's more cutting edge. T-cell receptor, TCR, diversity assays. Using high-throughput sequencing, we can now measure how diverse someone's T-cell or B-cell repertoire is. High diversity suggests healthy, naive cell pools, more adaptability. Low diversity, dominated by a few clones, suggests chronic stimulation or maybe CHIP. Some studies link higher TCR diversity to better health and less frailty in older age. It reflects immune reserve. Right now, it's mostly a research tool, not routine clinically, but it gives a deep look at immune aging.
And those inflammatory markers like CRP and IL-6, are they used practically?
Oh, definitely. Inflammatory markers like HS-CRP, high-sensitivity CRP, and IL-6 are probably the most accessible biomarkers for inflammaging in clinical practice. We know elevated levels are strongly linked to disability, mortality, and multiple age-related diseases, even in seemingly healthy elderly people. The Pulse Senior study showed this clearly. Higher IL-6 or CRP meant higher mortality risk. Successful agers had low levels. So clinically, if an older patient has a chronically high CRP, say over 3 milligl, without an obvious infection, it's a red flag for high inflammatory activity. It prompts looking for underlying causes, obesity, gum disease, etc. Some geriatricians track these over time.
And the CHIP testing you mentioned.
Right, the Chi-Chi panel. That's genomic sequencing of blood to look for those mutations in genes like DNMT3A, TT2, ASXL1, JK2. Finding cheat GP gives insight into hematopoietic aging. A 68-year-old with high-burden Chi might biologically have the marrow function and inflammatory potential of someone much older. This could justify more vigilant screening for blood count changes and definitely pushes for aggressive cardiovascular prevention. Again, routine screening isn't standard yet. It's done case by case, often in research or maybe in patients after cancer therapy.
Are there other ways to assess immune cells?
Yes, through other immune cell phenotyping using flow cytometry. This can measure proportions of naive versus memory T cells, expression of senescence markers like CD57, numbers of B cells, NK cells, etc. Research suggests patterns like a low CD4-CD8 ratio, low naive T cells, and high senescent CD8 T cells predict worse outcomes. It's not typically done routinely unless evaluating a specific immune deficiency, but it's common in specialized aging research clinics. And simpler things too, like checking immunoglobulin levels and vaccine titers. Measuring someone's antibody response after a vaccine directly shows how well their immune system can react. Clinicians might check tetanus or pneumococcal titers in infection-prone older patients. A poor response might trigger revaccination or further checks. And checking overall immunoglobulin levels, IgG, IgA, IgM, can rule out conditions like acquired hypogammaglobulinemia, which can happen in older adults.
So it sounds like researchers are trying to combine these into an overall immune age score.
Exactly. That's an emerging concept, creating a composite immune age score. Similar to epigenetic clocks, but for the immune system, using multiple parameters, T-cell ratios, cytokines, maybe TCR diversity, to calculate a single score that reflects biological immune age better than chronological age. These aren't ready for clinical prime time yet, but it shows the direction, quantifying how old someone's immune system really is. For Alex, simple starting points would be checking HS-CRP and IL-6 for Inflammaging, a complete blood count, looking for subtle clues like anemia, high MCV, low lymphocytes, and maybe CMV status. If infections are frequent, checking immunoglobulin levels makes sense too.
This has been incredibly detailed. Okay, let's bring it all together now. We've explored the science deeply. Let's translate it into action. Chapter 8. Practical strategies for maintaining immune function. What can Alex and all of us actually do?
Right, this is the crucial takeaway. What are the actionable steps? Number 1, absolutely top priority, is staying up to date on vaccinations. For older adults, this is non-negotiable. It primes the pump of their immune memory. And it's not just about preventing that specific disease. Preventing infections avoids that whole cascade of inflammation, muscle loss, functional decline that can follow a bad illness in an older person. A single pneumonia can really set someone back. So for Alex, annual flu shot, high-dosage Juventud, Shingrix, pneumococcal, COVID boosters, RSV vaccine, get them all.
Okay, vaccines first. What's next?
Optimize nutrition, especially protein. Sarcopenia, that age-related muscle loss, is directly bad for immunity. Muscles make immune-regulating factors and are amino acid stores during stress. Adequate protein helps maintain muscle and provides building blocks for immune cells and antibodies. Older adults often need more protein per meal due to anabolic resistance. Aiming for maybe 25-30 grams per meal is a good target. And broader nutrition, too, enough calories, vitamins, minerals like zinc and selenium. A malnourished immune system just can't fight well.
Nutrition, got it. How about exercise?
Hugely important. Regular exercise, both resistance and aerobic. It has potent immune benefits at any age. Moderate, regular exercise reduces systemic inflammation and can partly counteract immunosenescence. Studies show older exercisers have more naive T cells and better vaccine responses than sedentary peers. Resistance training builds muscle, good for immunity, reduces frailty. Aerobic exercise boosts heart and lung function, helping fight respiratory infections. Even just walking 30 minutes daily and some lightweight training makes a difference. Move it or lose it absolutely applies to immunity.
What else is in the practical toolkit?
Prioritize sleep. Chronic poor sleep raises inflammation and suppresses immune functions like NK cell activity. People sleeping less than 5-6 hours have weaker vaccine responses and get sick more easily. So, good sleep hygiene, aim for 7-8 hours. And treat issues like sleep apnea, which fuels inflammation. Also, maintain oral and periodontal health. This is often overlooked. Chronic gum disease is a persistent infection and source of inflammation. It's linked to higher systemic inflammation, even heart disease risk, and cognitive decline. Regular dental care helps quiet this source of inflammaging. Treating gum disease might actually lower IL-6.
Simple lifestyle factors, too.
Definitely. Avoid smoking and limit alcohol. Smoking is pro-inflammatory and damages lung defenses. Heavy alcohol use weakens immunity and harms the gut barrier. Quitting smoking and moderating alcohol removes 2 major immune stressors. And manage stress. Chronic stress blunts vaccine responses and flares up latent viruses. Techniques like mindfulness, social support, therapy can help reduce stress-related immune problems. Also, control comorbidities. Conditions like diabetes impair immune cells and raise inflammation. Good control of blood sugar, blood pressure, etc. prevents extra burdens on the immune system.
What about supplements or immune boosters?
Use prophylactic therapies only in specific high-risk situations and under guidance. Maybe vitamin D if deficient during a surge, or antivirals if exposed and high risk, but be very wary of unproven boosters. Critically, avoid unproven immune boosters. Many supplements make claims without evidence. Some can be harmful or interact with meds. Stick to the proven lifestyle measures. The boring basics really work in appropriate medical care.
And if someone does know they have CHE,
Then CHIP risk management becomes key. If Alex had known CHP, definitely ensure they're on a statin, counsel strongly on diet, exercise, maybe consider low-dose aspirin if cardiovascular risk is high. Possibly refer to clinical trials like for colchicine or IL-1, IL-6 blockers. If the CHIP clone is large or counts are dropping, see a hematologist. Testing isn't routine for everyone. Maybe consider it for unexplained early heart disease or odd blood counts. If positive, frame it as, okay, this clone raises inflammation risk. Let's be extra vigilant about heart health. If negative, general prevention still applies.
that's a really clear, practical roadmap. This whole deep dive has shown just how complex the link between aging and immunity is.
It really has. Time definitely wears down our immune defenses. That's immunosenescence. But it also, paradoxically, ramps up inflammation and inflammaging. Then you layer on things like shifts in the gut microbiome and those rogue CHIP clones in the bone marrow. It creates this perfect storm where the immune system struggles to regulate itself, leading to higher risk of infections, chronic diseases, frailty. Alex's fatigue and infections likely involve many of these layers. Fewer naive T cells, maybe latent CMV, maybe an unhelpful gut flora, potentially CHIP driving inflammation.
But the hopeful message is that this isn't all inevitable decline. Right?
Yeah.
It sounds like there's a lot we can influence.
Exactly. That's the crucial takeaway. Immune aging is modifiable to a real extent. Studies show it's not a one-way street. Interventions from senolytics to mTOR inhibitors to those fundamental lifestyle changes can partially rejuvenate immune function or lower that inflammatory burden. And we're learning that immune aging varies. Biomarkers might help us personalize interventions for those aging faster immunologically.
So for everyone listening, what's that essential immune maintenance stack distilled down?
Okay, the practical take-home stack for aging well immunologically is 1. Get all recommended vaccines, age-appropriate ones. 2. Engage in regular physical activity, strength and cardio. 3. Eat a nutritious diet, ample protein, plants, micronutrients. 4. Get quality sleep. 5. Maintain oral health. 6. Avoid smoking. Moderate alcohol. 7. Manage stress. 8. Keep chronic conditions in check. These things work together synergistically to support resilience.
And looking ahead to the cutting edge, what should people watch for with that strong note of caution?
Keep an eye on clinical research for things like rapalogs, low-dose rapamycin, targeted anti-inflammatory drugs for high inflammaging and maybe eventually microbiome transplants. And this is the critical FDA caution again, do not try DIY fecal transplants. It's dangerous.
Yeah.
Serious infections, even death have happened. Stick to proven, regulated medicine.
A vital warning. So, wrapping up, by understanding all this, by layering these strategies, we're helping the aging immune system do its job better policing the body.
That's the goal. Keeping infections down, vaccines working well, inflammation controlled, so that a 68-year-old like Alex, and all of us hoping to reach that age and beyond, can stay as healthy, energetic, and resilient as possible, living a life of continued learning and well-being.
Thank you for joining us on this deep dive into the fascinating world of immune aging. We hope you leave feeling not just informed, but truly empowered with these insights.